So you're able to comfortably hear me? Yeah, no problem. Venkat sir, I'm audible.
Yes, you are.
All right, perfect. Okay, great. All right. Okay, perfect. So now we are live on YouTube and also on Zoom. So welcome everyone. Uh, Thank you so much for joining in. And this is the month two of our regulatory Q&A series. Uh, I have been receiving a lot of questions from all of you, uh, obviously from also the past attendees of the session. And the common question that they asked was about little bit about information about clinical trials and investigators brochure, right? And how investigator brochures helps in clinical trial or clinical research, right? So as always, I'm joined by Venkat sir. Venkat sir brings 20 years of valuable experience in regulatory affairs. He has done MPharm in pharmacology. He's a proud pharmacist. He's an accomplished regulatory affairs professional and a senior regulatory consultant with over 20 years of experience in the pharmaceutical industry. His expertise comes from CMC, labeling, quality assurance, production, and a very, very strong track record in global regulatory submissions, especially with respect to Europe, US, and raw market. He's led a lot of regulatory strategies from early clinical phase trials to approval. He has held very senior roles in companies like Aurobindo, Sun Pharma, Parexel, Novartis, GSK, ESI, and Rosemont Pharma UK. He is very much regarded in the leadership in life cycle management in the companies. And thank you so much, sir, for being here and helping us every month to learn something. So welcome again, and thank you so much.
Thank you, Manu sir. It's my pleasure.
Thank you, sir.
Hello, hi everyone. Good evening. I really appreciate joining here, taking Saturday evening. So thank you so much. Yes. How many of you attended my last session?
Just give yes or no in the chat box.
Yeah,
Let's
in the
see.
chat box, please. Yeah.
If you have not attended, I'll send a link of that as well. Yeah, Dr. Nazia says yes. What about others? Anyone on YouTube attended the last session? Let us know. No, okay. All right, no.
Okay.
This one says no, Harsha says no, Aishu says no. Okay, so we have new attendees as well in this session. No problem.
Yeah.
I'll send you the recording of the last session so
Yeah.
that you can recap. This is a series we have planned. Venkat sir agreed to do this Q&A series every month. Specifically, we'll do either on Saturday, second Saturday, or second Friday of every month. And the objective is to create awareness about different aspects of regulatory affairs and clinical research. And today's topic is very close to all of our heart, specifically with respect to clinical research, what it is and what is investigators brochure. We'll start with that, and then we'll begin the Q&A. So if you have any questions with respect to clinical research, regulatory, please feel free to ask. This session is not about medical writing. So medical writing sessions happen on third uh, third of Fridays of every month. So please do join there and then ask questions related to medical writing or medical communications. This session is dedicated only for clinical research and regulatory affairs. All right. So are we good to go, sir?
Yes. Yeah. Right,
Perfect.
sir. Okay. Have you given permission to share the
Yes,
screen?
yes, yes,
Okay.
absolutely.
Perfect. Yeah.
Yes,
Let
yes.
me share my screen. Yeah.
Yeah,
So
please.
this I use it to present, okay, to write here as a board. Yeah.
Yeah, yeah, yeah.
So yeah, as you have already circulated that details, this session is mainly for a question and answer. So you please feel free to ask any questions you have in your mind,
Yeah.
whether you are already in the industry or not. Okay, so irrespective of that one, you can ask any questions. Okay, so this is a platform to clarify your doubts, or if you want to enter into industry, of course you can take a good understanding
Yeah,
from here. Okay,
absolutely.
so the subject, the session I will start. Okay, it's mainly the clinical phases. Probably most of you have this pharmacy background. you might be aware, okay, already different phases. So preclinical phase and sometimes phase 0, phase 1, phase 2, phase 3, and phase 4. So here the purpose of each phase, preclinical, it is animal studies or in vitro studies of any new drug before going into the human studies. Okay, so here we get good understanding about the drug, the PK/PD, and the toxicity of the drug. Okay, the safety we establish in animals first. Okay, so that gives a confidence to go to the next stage. So in some drugs, we start with there will be phase 0 as well. In phase 0, very limited subjects we take, okay, human subjects, and so here it gives more confidence, okay, to enter into phase 1. But it sometimes phase 0 or phase maybe there or may not be there. Next, in phase 1, we will establish mainly the safety and tolerability of the drug. Okay, that is the main objective of this phase 1 studies. Here the subjects are very limited, okay, maybe 20, 30, depends, okay, on the objective of the study. So here in phase 1, different experiments are conducted on these subjects to get a good understanding about the safety and tolerability of the drug. The effectiveness will be studied later, but first of all, safety is important. We know every drug, 3 parameters, as I always tell: safety, efficacy, and quality. Okay, these are the 3 parameters need to be established. So I think there are some students as well, probably you may or may not be aware of the regulatory process, okay, how the regulatory authorities will provide approval to the license. I think in my last class, I have explained what exactly for a drug, for example, risk versus benefit balance will be studied by the authorities before issuing the license. Okay, so once the license issued, then only the medicine should be placed in the pharmacy to sell to the patients, right? So how they establish, as we said, risk versus benefit. Okay, and here the drug can be studied in stage wise. Okay, the studies cannot happen all in once, right? So that drug, first of all, we need to understand the chemistry of the drug. Okay, then how it works in the body, okay, in the animals or in vitro studies on the tissues, etc. So there are different in vitro studies. Then once we understand that drug primarily, then it goes to the phase 1, right? Phase 1, phase 2, that means human studies. But in this human studies, okay, it is also progressive studies, right? That's why they made into different phases: phase 1, 2, 3. Right, in phase 1, safety and tolerability, and in phase 2, once the safety is established in phase 1, okay, so the minimal the dose, probably we don't understand thoroughly about the dose of the drug to be given. That will be studied in the phase 2, but we can get a good preliminary information. What is the safest dose to be given? Okay, is it a 10 milligram or 20 milligram from the animal studies and from the phase 1? Then when it goes to phase 2, we can understand the effectiveness of the drug. So where exactly this is effective, okay, and balancing the safety. For example, if any drug, if it is more binding nature, okay, if it is not metabolizing fast, then it is possible that the drug can reside in the body for longer time. That could lead to the toxicity, right? So this depending on the molecule nature, molecule characteristics, that and the results the showing in the ADME phase of the human, that phase 2 can be studied, right? Then from the phase 1 and phase 2 results, we can move to the phase 3. Here in phase 3, we can actually file the NDA, okay, new drug application. Phase 3, it is like a confirmatory studies from the phase 1 and phase 2. It gives, so it will be studied in the more number of subjects. So in more number of subjects, we get more understanding about the drug, right? So once the the safety, efficacy, and quality established and have sufficient knowledge about the molecule on these three parameters and a good confidence of this drug, then the NDA will be filed during the phase 3. Okay, so here the quality, that is CMC, we call chemistry manufacturing controls, more evolve during the phase 3. From the phase 1, we more concerned about the safety. In phase 2, we more concerned about safety and efficacy. In phase 3, we more concerned about quality because safety and efficacy already established in this one. Of course, in parallel we study, but the quality, the manufacturability, okay, the how the drug absorbs in the body, etc. Okay, in respect to the quality, we will study in the phase 3. Once we have got the license in phase 3, then what phase will start? Phase 4. Can someone tell me what is phase 4 called? What is the objective of phase 4? Can someone tell me, please?
Anyone in the chat box?
Like
Yeah.
interactive
PMS?
session. Yeah, please, respond to the questions here so that I will also be motivated. Yeah, I want to understand, the already the competency you have in this subject.
Yeah. So there are a lot of responses coming in, sir. One says Harsha says PMS. Mohammed Askar says PMS. Jashwant
PMS
says
means, sorry?
post-marketing surveillance.
Okay, PMS. Yeah. Okay.
Yeah. Jashwant says human trials.
Yeah.
Aishu says post-marketing surveillance.
Yeah. Excellent. Yeah. Right answer. Okay. Post-market surveillance and pharmacovigilance.
Yeah.
So here, once the license is issued and got approved, then it will reach to the more population, right? It will reach to hundreds of thousands of and lakhs of patients. That gives a realistic understanding about the drug in phase 4. And again, during that phase, of course, the research keep on continuing, right? Research will be continued. And then the PSUR, pharmacovigilance safety update report, will be prepared by the PVG team to submit to the agency, because that gives a proper understanding about the adverse events, how it is acting within the body, and it could reach to different races. Okay, maybe throughout the world, okay, there are different races available, and in each race the enzymes act differently, the hormones act differently, so the drug may be effective or may not be effective. So we could understand better during the post-market surveillance. In that post-market surveillance and pharmacovigilance, if the risk outweighs than benefit, then the agencies can still recall the drug. That means they may ask companies to withdraw that medicine because the risk is more compared to the benefit giving to the patient. So it could also be possible. Okay, this is how, okay, briefly about the phases. Now the main purpose here, my main objective of this webinar is to give you more of practical understanding what happens within the industry rather than just giving the slides and theory. I want this session to be more interactive for you to ask questions, to understand more and more, okay, and to enhance your knowledge. If you are studying still in the studies, probably you can connect your theory, pharmacology, whatever subjects you are studying, you can connect that one to this realistic practical experience. Okay, so I want to drive this to various websites, how it looks like and how to interpret those regulations or guidance available, okay, from the various authorities to enhance your knowledge here. Okay, any questions so far?
Participants, if you have any specific questions, please feel free to put in the chat box and we'll take those questions as we progress ahead.
So can someone tell me what is investigator's brochure? Do you know anything?
Anybody knows about IBs, investigator brochures? Let us know in the
limited
chat
knowledge
box.
is also fine. Yeah.
Yeah, don't worry about the perfect answer. There is no perfect answer.
Yes, exactly. Yeah.
You can put in the chat box again. If you want to speak, you can raise your virtual
Yes, yeah,
hand
please, yeah.
and I'll unmute you as well.
That is more
Yeah.
good.
Anyone wants to
Yeah,
speak, you can raise your virtual hand. Any idea about investigators brochure, what it is and why it is, what is the importance in clinical trials? If you do not know, type D and K in the chat box. D and K means do not know. And it's perfectly fine not to know. I'm waiting for some responses. Is it like ADR filing? Okay. Anybody else wants to give a try?
Okay. Right, no problem. So can you see the screen? I have opened here.
Yes, sir. We can see that. Kindly magnify it so that it's full screen
Yes,
and we
yes,
can see it
yeah,
properly.
yeah, perfect, yeah.
Yeah.
So
There are some responses coming in. One is it like ADR filing from
okay,
Mohammed Askar? Dr. Nazia says, inform investigators about the rationale, safety, and efficacy of the investigational
yes,
product.
perfect,
Harsha
yeah,
says clinical and non-clinical data.
yeah, exactly, yeah. So yes, rightly said. So this is including all non-clinical and clinical information about the drug that is going to be studied in the clinical site, and this is mainly meant for the health authorities and also the investigator, right? So first of all, we will see broadly what content we have in this investigator and how on what basis this investigator's brochure is made. That means the linking guidance and what exactly happens in the real world. Okay.
Yeah.
So this is one I have
Sir,
taken.
sorry, could you magnify it a little bit
Yeah.
or make it?
Yeah. This
Yeah.
is what is available. I have taken from online. Okay, public domain.
Public domain.
So it is not taken any confidential. So whatever is available in the public domain, I have downloaded this one. So if you see here, I think I have downloaded other one also. I couldn't see where I have downloaded, but it's okay. So this is investigational product Favipiravir. Okay, I think it is the antiviral. Okay, to my knowledge, this Ravir or something.
Yes, sir. Yeah, it is antiviral.
So yeah, so version 3.0. Okay, that means before this version there were two versions. Okay, they have made. So this is the third version. Okay, now this is in 2022. Okay, so this is the contents, what exactly it has, version 1, 2, 3. Okay, then contact information.
That part is called version control.
Yeah, version control, yes. That is
very,
important.
very important in regulatory and
Yeah,
medical
in
writing.
World, yes, exactly, yeah. Now, table of contents, you see here, summary, then introduction, then physical, chemical, and pharmaceutical properties and formulation, non-clinical studies, then effects in humans, summary of data and guidance for the investigator, okay, etc. So here, abbreviations, then changes to the previous edition, what changes they have made, adding relationship nature between ADL and AAL, okay, etc. Adding COVID-19 phase 2 clinical studies, okay. Changes to the previous edition, phase 2. So summary, this is the name of AAL. AAL has went through name changes, rebranding, okay, company, etc. They have given. Concluded the development of Favipiravir in any formulation different from 200 milligram. So this is the chemical name of the molecule. Then it is a broad antiviral drug with broad spectrum capabilities in non-clinical studies and has been investigated in clinical studies for its efficacy in uncomplicated influenza in adult subjects. Okay. Then physical, chemical, and pharmaceutical properties and formulation. So it is a novel nucleic acid analog that interferes with viral RNA replication. Okay, kept in dry area, stored at this. So this is about the chemistry of this molecule. Then pharmacology. So before I scroll further, I want to tell here. So the investigator brochure, this is a summary document for the investigator to understand about the molecule in detail. So any studies without a proper rationale, we should not conduct. So on what principle we should not conduct? What is the principle to be followed here? Do you know anyone? Do you know about this one? Anyone know about this? So clinical studies to perform, there must be a rationale, right? So where it is explained, in which guideline it is explained? As I said, it is not a structured webinar because I want to involve you as well in the discussion. Okay. Even if you don't know also, you can tell, yeah. So have you ever seen this guideline, good guideline for good clinical practice?
Yeah, its messages are coming in, sir.
Yeah.
ICH GCP.
Yeah, excellent. Okay. Excellent. So this is ICH GCP principles. So in the principles, if you see, can we go to the principle? Here there must be 13 principles, right? So as per the principle, without having a proper rationale, the study should not be conducted, right? That means the rationale should be explained in the protocol and investigator's brochure. Okay, then only the investigator will decide whether the ethics committee and investigator will decide whether to go ahead with the with the study or not. Okay, and to convince the investigator, first of all, the pharmacology should be in detail. How this drug is going to act in the body. That is pharmacology, nothing but the combination of kinetics and dynamics, right? So how this is going to act in the body, and then the drug, what it causes to the body. Okay, so once it is attached, for example, if it attaches to the receptor or if it interferes with some enzymes, whatever, that could cause consequential actions, right? So in the pharmacokinetics, the objective is to get that action done so that the patient can relieve from the disease, right? But when that has a consequences, it has the opposite reactions as well, right? What is that opposite reaction? Nothing but the adverse events. Okay, they are the dynamic effects. Okay, so it can, if it, for example, a drug meant to act on smooth muscle, so it may not act on that one smooth muscle, right? This can go to other muscles as well. So unnecessarily it could act on other receptors and throughout the body as well. So here the dynamics and kinetics we need to understand the whole pharmacology. So where we get the preliminary understanding? In vitro and animal studies, and then phase 1. For example, this one what we are seeing, it is a phase 2. That means they have conducted phase 1 and animal studies. So they must have a more knowledge already about the pharmacology. That means the safety and tolerability, toxicity, everything all might have already established. So now this phase 2 objective is to understand more about the effectiveness of this drug. What is the right dose to put, okay, to give to the patients in order to get that pharmacological response. Okay, so pharmacokinetics. Here I am not going to read, okay, each and every line. Of course, this you can read. So my recommendation is also, if you are a student, you should cultivate that habit also to read by yourself. Self-study is more important, okay? So don't think that to learn everything from the, from the teaching, okay? So you should learn by yourself as well, okay? So here, for example, plasma protein binding averages 53 to 54%, of which 65% is bound to albumin and 6.5% to alpha-1 acid glycoprotein. Okay, so this is the binding nature of this molecule. So when it binds, what happens? These proteins are carrier proteins, right? Some are carrier proteins. They carry the proteins to the cell, okay, wherever the target is required in order to in order to exhibit its action, right? So if there is no carrier, then there won't be any binding. If there is no binding, then the drug will be eliminated without any action. And then the metabolite, okay, metabolite is formed. This is how the conjugate is formed. Then it will be excreted predominantly in urine and to small extent into the fecus. In healthy volunteers, following administration of a single oral 400 milligram dose, 86.7% of the dose was recovered in the urine as this metabolite. Okay, that means the main kidneys are the main metabolic organ here for the elimination of this drug. So this is this pharmacokinetics where they have got from the phase 1 study. Now this they are writing in order to conduct phase 2 to give confidence to the investigator, right? This is the major metabolite. Then special populations, special populations like the pregnant ladies, okay, then who are breastfeeding ladies, or maybe some vulnerable patients like adults, okay, the aged geriatric patients, or if they already have a renal failure, then because it is excreting through renal mainly, so the renal failure patients, how this is going to be safe in those kind of patients. So the experimentation on the special population is also required as part of pharmacology. Okay, this is adding in the investigator's brochure. Then next, non-clinical data. So they have given here non-clinical information. Non-clinical in the animals, or it could be in vitro as well. So here, this Noel in monkeys, okay, they have studied. Noel was 100 milligram per kilogram per day following 14 days of treatment they have given here. So this 100 milligram per 1 kg dose they have given. So this gives some understanding about the determining the dose in humans. Okay, so here for 1 kg body weight, 100 milligram given for the monkeys. So proportionately, how much mg to be given to the body, but here there are different calculations. Okay, in the clinical studies, clinical scientists will decide based on the body volume, surface ratio, then they will always go less 10x for the humans. Okay, so this will be determined by the clinical scientists based on the protocol, and they will provide in the protocol. Okay. Then the fertility effects in rats was studied. Okay, so clinical experience in influenza. Okay, so particular to influenza, what they have studied, overall 40 clinical studies with favipiravir has been completed and analyzed. Studies designated as US were designed and conducted primarily in the US. Studies designated as JP were designed and conducted in Japan. Okay, so here let me understand here what is the objective summary here. Okay, what they wanted to establish here. If we go to the contents here, so summary, okay, introduction, non-clinical effects in humans, safety and efficacy, marketing experience, summary of data and guidance for the investigator, usage, doses and administration, etc. They have given phase 1 single safety and efficacy, phase 1 single dose and multiple dose, phase 2 studies, phase 3 clinical overall summary of safety from key clinical studies. Okay. Okay, let me go through this one. I'm also reading this document, along with you. Then clinical experience in influenza, particular to the influenza, they have studied. Here they have conducted one experiment and they have drawn the data. Then the clinical safety. Okay, again here a 5-day regimen of favipiravir was well tolerated when administered to adult and elderly subjects. The safety profile at the intended influenza dose, 1800 milligram, okay, one single day 1 followed by 800 milligram day 2 to day 5 is broadly comparable. Then clinical experience in other RNA viral diseases. Okay, they have extended this study to not only influenza, other viral diseases as well. Okay, that is when they establish something here, they can extend the indication. Okay, I will show you other document how different indications will be written. Then introduction, this is a novel nucleic acid analog that interferes with viral RNA replication. Okay, so it is going to act on the viral RNA replication that will stop the replication of the RNA. That's how the protein of this virus will be stopped, right? Then physical chemical. So this is my main area, my main experience. So as I said, CMC quality, my strong experience. So the physical, chemical, and pharmaceutical properties of the dossier, I will be involved here. Okay, the molecular weight is 157. Then the inactive ingredients, how the tablets are made, mixing with different excipients, then the stability, etc. they have given. So in phase, this is a phase 2 study, right? So still the CMC will be not that stronger here. Then chemical structure, storage and handling, manufacturers, active substance manufacturers. Okay, so these are from India. Then finished product manufacturer, Strides Pharma Science Limited, non-clinical studies. Okay, so that means this is already a marketed product. Okay, this is I hope you know the difference between innovator and generic product. So this is a generic product. They have performed phase 2 study in order to establish how their particular product is effective in certain patients. Non-clinical studies, so they have conducted non-clinical studies as well. Then mechanism of action, so this is pharmacology. So the mechanism of action, here you must have a subject like anatomy, physiology, right? So you should have a thorough understanding of anatomy and physiology in order to understand the mechanism of action. It is a nucleic acid analog that is ribosylated and phosphorylated within the cells, forming T705 ribosyl monophosphate, which can be further phosphorylated to the di and triphosphate analogs. As with other nucleoside analogs, studies suggest favipiravir interferes with viral RNA replication. Initial studies demonstrated that this one, but not of favipiravir inhibited influenza viral RNA synthesis by viral polymerase in vitro at a low micromolar concentrations. Okay, so this is how, if you want anyone to understand more about the mechanism of action, because I have studied for few drugs, this drug is new for me. I have taken whatever the document available in the public domain, I have taken to explain you how the actual investigator brochure looks like. Okay, if you want to understand more of this mechanism of action, let me or Manoj sir know, so we can definitely will explain you. Yeah, maybe separately also. Yeah,
Absolutely.
Yeah. Then in vitro and in vivo bioactivity in influenza. Okay, so they have studied both, and here nice picture, probably you try to understand more. Okay. Again, you can connect to both of us. We will also learn as part of, while explaining to you. Then secondary pharmacology, of course, the next stages, once it is the first primary pharmacology and what consequential on what it acts. For example, was tested for off-target reactivity in vitro radioligand binding assays. So this is one kind of study, in vitro study, against 25 purified receptors, including CYP19, steroid 5, alpha reductase, androgen, etc., etc. And they come to know pharmacology of this drug more. Okay, safety pharmacology, then pharmacokinetics and product metabolism in animals. Then toxicology, single dose studies, repeat dose studies, rats, dogs, monkeys. So these animals, again, why these animals, why not other animals? This will be depending on the kind of similarity. Okay, the mechanism of action. Once we understand about this molecule, then the clinical scientists, they will decide which animal is appropriate to study the pharmacology. Okay, because for example here the RNA enzyme, right, it is working on the mainly this RNA. So these monkeys or dogs, etc., that gives us a good similarity, okay, to the humans. That's how they have selected these animals. Then genetic toxicology, how it is going to affect on the genetic, okay, within the human body. Then reproductive and developmental toxicity. So you probably aware there are some drugs they don't recommend when lady is pregnant. So that will be part of this. It will be included in investigators brochure. Fertility and early embryonic development studies. Then early embryonic antiviral comparison study, embryo fetal development study, rat embryo fetal development study in mouse, then rabbit, monkey. So probably they might have not studied everything, so they can take from the literature as well. As I said, it is an innovator molecule, so they take some of the information from the literature where it is not required to do again. Okay, that's how generic drugs supply with affordable prices, right? How? Because they won't conduct many of the these clinical studies. Okay, pre and postnatal development study, then reproductive toxicity studies with this metabolite specifically, then testicular toxicity. So usually this IB is around 60 pages to 200 pages long. Okay.
Yeah.
Then local tolerance studies, other toxicity studies, effects in humans. Okay, 6th section, pharmacokinetics and product metabolism in humans. These are the different parameters. Cmax, if you are a pharmacy student, probably you know a little bit what is Cmax, Tmax, area under the curve, okay, half-life. If you want to tell something about this voluntarily, please raise your hand and tell. Okay, it is a clearance. Okay, this will tell about how renal clearance is this one. Then okay, dose excreted, Cmax, etc. So this one for main drug, and this is for the metabolite. They have studied again all these parameters. Okay, as we know that this drug is converting into metabolite. Okay, so that means when in the body, once we have taken, both forms will be available in the body. Drug interactions. Okay, so if we know that we might have seen in our families as well, we will take multiple drugs, right? So sometimes food interaction also, some drugs should not be taken with the high fat meal, and some drugs should be taken with the high with the high and without high fat meal. So both scenarios could exist. So we need to establish which one is appropriate for that drug. and also how that is interacting with other drugs. For example, acetaminophen in combination with acetaminophen increased acetaminophen blood levels 14 to 17%. So that means the doctors will tell to the patients, so be cautious when they are taking, along with acetaminophen for other indications. So hydrolazine, we know it is, I think, it is renal, what you call, for blood pressure, I think. Okay, it is used along with the blood pressure drug. So result in a 13% reduction. So that means the effectiveness of this hydrolazine is reducing when we are taking combination with the favipiravir. Okay. So this is how different studies, okay, with various drugs. So how do they come to know? Again, they have to conduct the study on the human subjects. All right. Safety and efficacy, phase 1 single dose and multiple dose studies. Phase 1 completed in Japan, phase 1 completed in Japan. So different study designs. So the design is important. The reason is why design is important in order to achieve the objectives of the study. Right. So if the design is not appropriate, then we will get the results, but that will not be useful. Okay, for what object? For example, here you want to study how the drug interacts with the hydrolazine, right? So co-administration of this both the drugs, what will happen? For example, you have designed, okay, with some similar drug. Okay, not if hydrolazine is not available in the market, you have taken some other drug, then you cannot achieve the objective actually what you want to do. Or maybe with another analogy also. For example, if you have taken only some 10 subjects, okay, to study this one, you have designed the study maybe wrongly designed, okay, not wrongly designed by the clinical scientists, just to study in the 10 subjects, that will not give accurate information how this is going to act. So maybe 30 subjects are required in order to study this one. So they have to, based on the statistics, based on their literature and everything, they will take into consideration in designing the study. Okay. Then you see here, what are the efficacy and safety outcomes they are expecting from the PK, etc., etc. Okay, they have given clearly here. So each phase 1, but different studies, okay, for different purposes. Okay. Then you see all these are different studies. Still we are at the 35 page number. I hope I'm not boring with this explanation, but here as I said, my main purpose is to give you bit more understanding rather than just going through the slides. Okay, feel free to ask questions at any time. Phase 1 completed, yes. Okay, all these they have captured here. I'm not sure whether you are aware of clinicaltrials.gov site. Okay, there you can see all the clinical trials happening on any drugs. Okay, from that site. Just text me. Okay, text Mano sir if you want to know about that one. So I will share that link as well. Then completed phase 1 studies. Okay, this is they have summarized phase 2 studies. 3 phase 2 studies in subjects with uncomplicated influenza have been completed. So here, phase 2, they wanted to particularly study for the influenza. So in Japan, they have done these studies. Phase 2 completed, South Africa, Australia, New Zealand, and Latin America. Okay, etc., etc. Phase 3 studies, uncomplicated influenza. Okay, for 4 phase 3 studies, they studied in subjects with uncomplicated influenza. They have studied again in Japan, okay, in different countries. So here COVID-19, phase 2 study in subjects with COVID-19 has been completed. So particularly for COVID-19, They have run this phase 2. Phase 2 completed. Phase 3 study. Okay, in Japan, clinical and virological efficacy from 3 key studies. Clinical efficacy from 3 key studies. Accelerated failure time model. Mean time to elevation. Right. Overall summary of safety from key clinical studies. Safety in the population of subjects with confirmed or suspected influenza treated with a 5-day regimen of favipiravir at the proposed dose was assessed using data from the 3 key studies. Okay, these are the studies they have mainly conducted in this investigators brochure. The safety population was defined as subjects who had received any amount of study drug. The majority of all TEAs in these subjects have been mild in severity and assessed as unrelated to study drug and resolved without sequelae. Most frequent adverse events from the 3 clinical studies. These are the adverse events they have observed: gastrointestinal infections and infestations, investigations, nervous system, respiratory, vascular disorders. These were the main adverse events they have observed, and the severity also they have. Number of N means number of population, the subjects they have taken 894 and here 1653. Serious adverse events, nothing. Unrelated serious adverse events they have observed in 5 and 4. So that means they need to update their investigators brochure for the next version, keeping these serious adverse events, okay, into the list. So the adverse events are 2 types, right? Expected and unexpected. Okay, expected, the investigator and the site will know about the expected adverse events. They will be prepared for the risk the subjects have. But unexpected, which is something they don't know, they don't know that this drug is going to cause this adverse event. So in that case, they have to go back to their study of pharmacology, etc., why this situation has come, and they need to study further, pharmacology about that one. Of course, once they have observed adverse event, they immediately, there is a procedure what to follow, and they need to be informed to the health authority. Okay, within sponsor they need to inform, and immediately to the health authority and others as well. Yeah. Then effects on uric acid. Okay, we are still at page 60. Baseline summary of uric acid observed. I think we have time, right? Yeah, 8:40. Summary of data and guidance for the investigator. Okay, this is the guidance for the investigator. He says is a selective nucleic analog that is converted to this one by cellular kinases. Favipiravir, I'm feeling very difficult to pronounce this one. Developed for the treatment of acute uncomplicated illness due to influenza infection in patients 18 years of age or older. Okay, etc., etc. Dosage and administration. This is so. These are the recommendations they have given to the investigator. And this, along with the investigator's brochure, the sponsor will train the investigator also to go into thorough of this investigator's brochure. Okay. Drug interaction. So this is all part of ICH GCP principles. This is again 86-page document. If you read this guidance document, you will clearly understand, okay, where this comes from. And this is the investigator responsibilities, etc., etc., how communicate with IRB, IEC, etc. Here in annexure, you can find, okay, the structure. So here in the ICH guideline, they have given appendix investigators brochure. From this, you can understand introduction, development of the investigators brochure, reference safety information. So these are the sections mandatorily. This is the template the sponsor has to follow in order to prepare investigators brochure. So who will prepare investigators brochure? Can someone answer now?
Yes, participants. Now, so much of discussion about it. Now, can you answer that?
Participate, yeah, whatever. Whether it is the right answer or wrong answer, you can tell based on the understanding what I have told now. So as per your understanding, who is going to prepare the investigator's brochure now? Kindly type, yeah.
You can type in the chat box, type DNK if you still didn't know.
So, yeah, regulatory writer, yes, will be involved, but it is prepared by the cross-functional teams. So, okay, clinical teams, non-clinical, regulatory, medical,
Yeah.
what else, pharmacovigilance, they all be involved, okay, in preparing this one. But the regulatory, in the end, they will take care of the format, right? If the format is not in line with this ICH guideline, then the agencies or won't take, and it is also non-compliance, right?
Absolutely.
So it should be complied to this template that will be taken care by the regulatory quality will be involved in the CMC data, as I said earlier, and also regulatory clinical to put it in a right format. Okay, so everyone will be involved. It is a cross-functional document. Yeah, the clinical trial protocol and protocol amendment. Okay, so this protocol is detailed how to conduct the study. Okay, what exactly the trial design, everything will be explained here. Okay, so that is a kind of a summary document and it is a detailed one. I think this is the main objective for us today, okay, to present briefly. So here I will take few more minutes to explain. Now, as we discussed, there are different phases. Clinical phase development, go into phase wise progressively, phase 1, phase 2, phase 3. In phase 3 we will get the license, then it goes to post marketing pharmacovigilance, that is a phase 4. So let's assume, okay, based on the satisfactory clinical studies, the authorities, health authorities issue the license to the particular company. Okay, whatever you take, Aurobindo Pharma, Reddy Labs, okay, some drug, Ramipril or whatever. Once you have got the license, do you think this investigator's brochure to keep updated all the time?
Type yes or no in the chat box. Let's see.
Is it going to be exist after license is issued?
Dr. Nazia says yes.
Okay. So, but the answer is no. Okay. So from the investigator's brochure, we are going to conduct the studies and we will get the data, and this data is going to be transformed into a product manual. For example, iPhone, right? During the design, they will discuss about the protocol. How much is the screen? Is it handy? What is the weight of this one? Okay, what is the camera they're going to put? And how, if you take the picture, how it is going to be appear on the screen? Is it going to be vertical or horizontal? So the design. So during the design, they will keep developing understanding about the product. So here the investigator's brochure, and from the results, we are going to understand about the product more and more, right? Each stage. So once we have got a thorough understanding, then we will be confident. That's how we will get the license. That means we know about our product. At least we have come to a stage, we believe that the product is going to be safe and effective and quality in the patient. It is going to be work properly for that particular disease. Maybe we will come to know more information, new information. But the thing is, so far, based on the confidence the agency has given us, now we have to present our manual to the healthcare professionals. Healthcare professionals are nothing but doctors, nurses, or whoever. They want to understand, for example, now the medical marketing team, they will go to the doctor, they will try to explain, we have developed this product Ramipril, And if you Ramipril, if you prescribe to the patient, this is going to decrease the blood pressure. Then doctor will ask the questions. Tell me, what are the studies you have conducted? What is the mechanism of action? Where it is happening? Can I give this tablet to the renal failure patients? Can I give this tablet to the pregnant ladies? So he will ask for the information. For that information, he cannot go to the clinical studies. But where he can reach to that information, that is nothing but we call it as product information. That is a manual for that particular product. So now I'm going to show you that one, how it looks like. That is available not for the same product, but different product I have taken. Okay, my favorite drug Ramipril, because it is easy for me to explain. Okay, most of the times, yeah. So this is Australia authority. They have given the approval for Ramipril for a Sandoz company, and this is the product information of that particular drug. As I said, it is a manual. Okay, it is a brochure for that drug, whatever name you call. In Europe, they call it as a summary of product character information, summary of product characteristics. In US, it is called product information. And in Canada, we call it as product monograph. And here they call it as product information again in Australia. So different names, okay? So this is meant for healthcare professionals. So they have conducted the studies. Now we can see how this is translated, our clinical and non-clinical information, how this is translated here in the product information, right? So therapeutic indications. This drug is meant for hypertension. This is also meant for heart failure. This is also meant for progressive renal failure in patients. This is also meant for myocardial infarction. It is also for this one. But dosages, the posology may be changed. But these are for many indications, 1, 2, 3, 4, 5. Sometimes you might be surprised. The patient may not have the hypertension. Then you may get a dilemma. Why this is given? Because it is for, he might have got the stroke or some infarction, myocardial infarction or something, etc. Revascularization procedure, etc. Or in diabetic patients. Even in that condition also, the drug can be prescribed. But how they have come to this conclusion? From the clinical studies. That means they have studied in some subjects about this one, how it is going to. That study, again, investigators brochure, they have justified to conduct that study. And they have included that indication. They have got good results, safety results. They have included that indication here. Dose and method. Okay, so the dose will be determined in which phase? Dose will be determined. Please respond, yeah. Whether it is wrong or right, don't bother. Okay, everyone. Okay, we all know nothing. Okay, when we have started, we learn something. Okay, learn in the process. So where do we understand how much dose should be given? In which phase we can come to know? I have
participants. You want to answer that?
In my, in the beginning I have told. No, it is in phase 2. In phase 1, we understand about the safety, and in phase 2, we understand about more of effectiveness of the drug.
Effectiveness and side effects.
Yes, exactly. Okay. So then it is good. Thank you for responding. Yeah. Then post-MI heart failure, doses in patients at increased cardiovascular risk. Okay, so this, you see lot of information here. This is all where they have got this information. They cannot get from just like that, right? They have to study, they have to prove, and then only include, then explain to the doctors. Okay, then give to the patients. Okay, so patients will get relief from the disease. So this is all stage wise, yeah. Contraindications. So you see, hypersensitivity to Ramipril, history of heredity, then pregnancy, lactation, renal failure. So what exactly they have to do, they explained here. Then special warnings: angioedema, head, neck, extremities, intestinal, hypotension, patients with hyperstimulated renin-angiotensin system. Okay, so kindly go through yourself. Okay, I will leave it to you to further study, right?
And we'll share these documents.
Yes,
Don't worry
yeah,
about that. We'll
yes, I
share
will
them.
share all the
If you're
documents.
watching this on YouTube, in the description of the YouTube video, the links of this documents will be shared. If you are on Zoom and if you have registered earlier, then you'll get an email with the document details. Okay, don't worry about that.
Yeah.
All
Then
right.
fertility, pregnancy, and lactation. Okay, so these are special populations, right? They have studied this one again in these kind of patients. So effect on fertility, no data available. That means they have not studied this drug. So doctor, if he doesn't know, they should not give confidently to these pregnant ladies. Okay, use in pregnancy, category D. Okay, as with all ACE inhibitors, ramip must not be taken during pregnancy. Okay, so that means it is strictly prohibited to give to the pregnant ladies. Use in lactation, effects on ability to drive. So next question is whether can we drive taking this one. Doctors will take, okay, for example, sedative drugs, okay, then they will say that don't take only in the night, or don't go to work or whatever, drive, etc. Okay. So adverse effects, more common. So adverse effects, that is divided into various categories, right? More common. So immediately after ramipril, it is very common. Dizziness, weakness, nausea, headache. This is very common. They will appear. So immediately when doctor will give ramipril and ask patient to see again within 1 week or 2 weeks, patients start complaining, sir, after started taking this one, I am getting a lot of headache and all. Then doctor come to know that it is a part of adverse event. There is nothing to worry. Okay. Then less common. Okay, these are less common. So it is, that means the doctor will pay attention if the less common the patient get, right? That means they need to see thoroughly whether drug is, maybe his liver is not working properly or his kidney is not working properly to metabolize, then he's getting more toxicity. That toxicity is leading to these problems. So he will change to other drug. Okay. Like this. Okay, then overdose. What should you do? For example, if by mistake patient has taken 2 tablets instead of 1, etc., etc., then pharmacological properties. So here pharmacodynamics and pharmacokinetics. So here mechanism of action. So how, first of all, the mechanism of action, just can you guess when this mechanism of action can be established? Just guess.
Which phase?
Which phase, yeah.
Yeah, which phase?
A preliminary understanding about the mechanism of action, where we can come to know. This is my exact question.
1, 2, 3, 4. Which phase? Just put in the chat box. Number 1, 2, 3, or 4.
Just a guess.
You can take just a wild guess. Don't worry. There is no right or wrong answer here. Anybody in the group? Let's see. Preclinical studies.
Yes, very true. So preclinical, we will come to know, okay, the basic understanding how it is. Yes, in phase 1, we'll know more and more about this mechanism of action. So here in animal studies and in vitro studies, we'll come to know how this is, the drug is, combining with what enzyme. Okay, angiotensin 1 enzyme. Here it is combining, and this is stopping the other, consequential reactions. Okay, that is how here the main, that for example, we need to understand, probably I can recommend the pharmacy students also to understand more and more about the disease pattern in various therapeutic groups. Okay. For example, blood pressure. There are different factors which causes blood pressure. In some it is mainly the vasodilatation and vasoconstriction. It is causing this one. And in some patients because of renal problems, and in some patients because of other cardiac issues or something. So the genesis of this blood pressure should understand more. Okay, before, so doctors will come to know from diagnosis and from their experience what exactly the genesis. Okay, for example, if it is a obese patient, then they will expect, okay, something like more vascular, then constriction might be. Or if it is already thin but some problem, then he will study more about his kidney problems and all, okay, etc. Then, for example, if the patient has kidney, some other problem, okay, not because of vasoconstriction, some other kidney problems, then doctors will not recommend this ramipril. They will go for other drug.
Yeah.
Okay, they will go for other drug. 1 second, sorry, there is some interruption.
All right, participants, how are you finding this session? Type good, bad, or ugly in the chat box. Let's see. What did we learn so far? We understood about clinical trial. We understood about investigator brochure. We understood about the product information. We understood about the ICH E6. We understood all of these 4 important aspects, though it was not initially planned, but then as the experience speaks, a lot of information starts coming in, right? So that's the advantage of having Venkat sir for this session. Venkat sir, you want to take a pause and have a glass of water quickly?
Yeah.
All right. So he clearly explained about the importance of clinical trial, right? A clinical trial is a research study which is conducted in human participants to evaluate the safety, effectiveness, and the side effects of new drug treatments or medical devices. Medical devices are also, part of clinical trials, right? And then clinical trial typically contains different phases. So we have phase 1, where we test the safety and dosage in a small group of healthy volunteers mostly. Then we have phase 2, where we evaluate the effectiveness and side effects in more number of patients. Then phase 3, where we confirm the effectiveness in large populations before the drug goes for regulatory approval. Then we have phase 4, which monitors the long-term effects of the drug after the drug is marketed, right? Because why? Because during the clinical trials, we have a limitation with the number of participants, right? So post-marketing, monitoring is absolutely important, and that is where even the pharmacovigilance activities come into picture here, right? So these trials, the 4 different trials, 4 phases of these trials, they are very, very essential in bringing the new medications and the medical devices from the lab to the patients, right? They're very, very important. And then we understood about the investigator's brochure, which is a very comprehensive, detailed document that provides investigators and health authorities with essential information about drug which is being studied, right? And what does it contain? It contains everything. It contains the non-clinical data like pharmacology and toxicology studies. It explains about the clinical experience and the safety information. Then it also explains the scientific rationale for conducting the human studies, right? It also, and then this entire thing is based on the ICH E6 principles, right? As Sir rightly said, everything in the regulatory medical writing or the regulatory affairs has to be as per the guidelines. There is nothing which is not done as per the template. So the final authority who approves these documents is somebody who's coming from quality and who understands these guidelines, right? And then IB serves as the very important reference for investigators to understand the drug profile and the clinical trials. That is where the product drug profile comes into picture, which Sir is explaining right now, Ramipril's product information, right? So thank you, sir. Please continue.
Yeah. So here we are at section 5 now, pharmacology. So here dynamics and pharmacokinetics. I have already explained to the other drug. Probably I will leave this to you, okay, to read and understand more on this one, okay. And each subject is different, okay. Each patient is different, so he might already have some other, problems, etc. So they need to give a combination drug or etc. So here different studies are required, okay. These are the CMC sections, okay, the conditions, stability, etc., etc. Okay, that's all. This is product information. So I will stop here and I will take question and answers here
Yeah,
from the
absolutely.
participants. Yeah.
So thank you so much, sir, for explaining in such a detailed manner of all these four important aspects which are part of clinical trials. Thank you again. So participants, as you know, this series we are planning to answer your most common questions on regulatory affairs. And this session specifically was based on clinical research and these guidelines, right? And the IB and the ICH and all of that. So if you have any questions, please feel free to ask now. You can simply put in the chat box, or if you want to unmute and talk, that's perfectly fine. Those of you on Zoom, you can now unmute and talk. You can ask your specific question. So can we stop sharing the screen?
Yeah.
Perfect. Any questions, guys? Please feel free to ask. All right. So
And
we're
any
here for
suggestions
some.
also for
Yeah,
me, let me
suggestions
know.
also. Yeah,
So for the
next
next,
session.
so if you really want PowerPoint slides, that is going to be helpful. Let me know.
Yeah.
If anything to be changed, okay, from my side, let me know, please. Yeah.
Yeah. Yeah. So let's go ahead with questions. If you have any specific questions, feel free to ask in the chat box, or you can also unmute and talk. All of you can now unmute if you click on Zoom. There is an option to speak. You can simply go ahead and speak.
Yeah,
Those
but I
of you on
expect more
YouTube,
participation. Yeah.
yeah,
Okay.
yeah.
So I don't know
Please
how
do
many of you are
participate.
already in the industry. So, they like more, speaking guys. Yeah.
Yeah, absolutely.
Communication is the
So
key.
those of you in industry, type I in the chat box. Those of you in academia, type A in the chat box. Academia means both teachers and students, right? So let's see how many of you are from industry and how many of you are from academia. Can you guys please interact? It makes clinical pharmacist. Okay.
Okay, good.
Jashwant says B. What is B? Is it academia or industry?
Maybe industry, I think. Just
Yeah.
one. Is it industry? Just one?
I is industry. B is A is academia. All right. Industry, yeah. Dr. Nazia is from industry. What about others? Any questions you have with respect to regulatory affairs? Now you may ask that. If you ask, we'll continue these sessions. That's why these Q&A sessions are supposed to be, right? I don't see any questions on YouTube.
Hello, sir.
Yes,
Yes,
Adnan, go ahead.
So I would like to, ask you, from working into the clinical sector, and how can a person, working as a clinical pharmacist and then work into the regulatory affairs, like shifting it from clinical to non-clinical.
Shifting from clinical as a clinical pharmacist to regulatory. That's what your question?
Yes, sir.
Yeah. Okay.
Yeah. Okay. Adnan, may I ask you few questions? Okay. How many years of experience you have as a clinical pharmacist?
So actually, I've recently joined as a clinical pharmacist.
Okay. Okay. What is your day-to-day work as a clinical pharmacist?
I actually work direct into the patient care. I do regular auditing of the prescription according to the NABH guidelines, and I also, counsel the patient whenever I go to the ward rounds, and to take some histories of the patient, and to evaluate all the things, and even in the medication part as well. Like any drug interactions are going on, any therapeutic duplications, and also trying to reduce the hospital cost to the patient, overall the cost, by reducing their medications part,
Okay.
unnecessary, or we can say it as a polypharmacy.
Okay, you will review their prescriptions in detail, right?
Yes. Yes.
Okay, and you suggest any reduction is possible in the number of medications,
Yes, sir.
or maybe any drugs can be removed, etc. Right? Okay.
Yes, sir.
So
And also, yeah.
do you study any drugs specifically, or do you see the patient overall?
So, whenever I get time, I also learn about medication part as well. Whenever I
Okay.
get time
Okay.
for myself.
Yes. Yeah. So here, you are already dealing with different medications to the patient, right? That means how you are able to review different drugs. That means a drug-drug interaction you are studying, okay, you are understanding. So this is all part of that clinical study. Okay, that
Yes, sir.
means the basics and all you are already familiar. Okay, so if you want to switch to the the clinical regulatory. Okay, of course, quality regulatory again, yeah, what I am doing, that is a completely different one. Okay, so here manufacturing process and analytical studies, then stability. Okay, all you need to understand. Okay, that is a different area, but you can easily connect it to this clinical and non-clinical regulatory. So here the main thing is the technicalities, okay, technical understanding. For example, any disease, okay, genesis and their the pattern, etc., you already know, okay, to some extent. Here you need to understand how this is connected to the clinical studies, what exactly they do in the clinical study. So if you learn about that one, then you can easily switch to the regulatory.
Okay, sir.
That
Thank
means
you,
you
sir.
need some training,
Yeah.
okay, and then you need to enhance your CV, what you have learned from the training, and you need to show in your CV, okay, what are your strengths, okay, how can you easily switch from the current job to that particular clinical regulatory. I recommend you, if possible, take training, or maybe you can self-read also from the public domains. For example, the one I have shown, investigators brochure, ICH guidelines, and this product information. You can write, you can put everything this in the CV. Okay, familiar, to review the product information, familiar to understanding about this investigators brochure. Okay, you can keep all these details in the CV and you can try, or maybe you can join as an intern. Okay, then probably you can enhance your knowledge. Yeah.
Thank you, sir. Thank you. It was really an amazing session. I really enjoyed.
Thank you, Adnan.
Perfect.
And
Thank you, Adnan. Yeah. And if you're in
thank you.
Hyderabad, please connect with Venkat sir on LinkedIn. He is also in Hyderabad and definitely will
Yeah.
guide you more there. Thank you. All right.
Yes, sir. Sir, actually, I'm from Gujarat, Surat.
That's
No
perfectly fine.
problem.
You
Yeah.
can still connect on LinkedIn and discuss more.
Yeah,
No
we can
problem.
connect. Yeah, yeah.
Sure, sir. Sure,
Distance is
sir.
not an
Thank
issue
you.
here. Yeah. We'll all
Yeah.
try
Dr.
Thank
to
you, sir.
Najia
support you.
from, yeah, sorry. Sorry to interrupt. Yeah.
Yeah, you said.
Dr. Najia, are you from which industry? Is it okay for you to tell? Medical writing.
Medical writing outsourcing. All right.
Yeah, good to know. Yeah.
That's interesting. Yeah. Perfect. Any other questions, guys? Let us know. If there are no questions, type NQ in the chat box. That will tell us that you don't have any questions and we are good to go. And we'll meet again next month for these regulatory sessions. The medical writing session will happen next week, one on Wednesday and one on Saturday, next Saturday. So that will be on 19th and 20th. 19th, we'll discuss about scientific writing in medical writing, the role of scientific writing in medical writing. And on 22nd, we'll understand about the role of freelance medical writing. These are the two sessions scheduled on next week. So stay tuned. You'll find more details on my LinkedIn update. And if you're part of the WhatsApp channel, then you'll also get the information on WhatsApp itself. So stay tuned. And thank you so much for joining in for these sessions. If you have any specific query from Venkat sir on regulatory, from me on medical communications or medical writing, please feel free to connect on LinkedIn. Or if you know our email IDs, you can also send an email and we'll discuss and we'll come back with more of these sessions. If you have any suggestions or feedbacks, don't worry, you can also send an email or WhatsApp me. Some of you have my number as well. So you can WhatsApp me the next topics and we'll try to take up those topics in the upcoming sessions. So anything related to medical communications, I'll take care of it. Anything related to regulatory affairs that happens every month, Venkat sir will do that. So for us, like Venkat sir has voluntarily agreed to be part of these sessions and share as much knowledge and information with all of us to help all of us grow better and better in regulatory domain. All right, so stay tuned for more information. And if you want to download the certificate of participation for these sessions, then again, the details will be sent out to you, or maybe let me just quickly share that information with all of you right here. So you can always download a certificate of participation for these sessions. And this is what you need to do for this. I'll share my screen and quickly explain. Is that okay? So let's go ahead. So this is this portal that we have created for sharing knowledge and helping all of you access these informations, right? And if you go to these courses, you can go to this session here, which is questions and answers in regulatory affairs. This is what the monthly session is all about. And you can click on this button called for free. It's a free course. And all these sessions will be updated here. The last session that happened on October 17th is available here. You can watch that. And the next session, that is today's session, will be updated here by today in another 1 hour's time. So after registering, you'll be able to download a certificate of participation. This certificate of participation will remind you about these sessions and also help you show that in LinkedIn on different other platforms. All right. And this will continue, or we'll keep adding more of these sessions going forward. Based on your suggestions, we'll keep adding a lot of topics in regulatory affairs as well. Okay. So thank you so much for being here with us. And Venkat sir, thank you so much for being an expert speaker on all these sessions and sharing so much of information, which is not possible for us to learn otherwise, right? There are thousands of rupees of courses for these kind of sessions. So you have agreed to share all of this for free to our participants. Thank you so much for that.
Sir, it's my pleasure. Yeah. I wish you all good evening. Okay. And good luck in your futures. Yeah. Thank you.
Thank you. Thank you so much, sir. And thank you so much, participants on Zoom and YouTube. I'm closing the session on YouTube. We'll continue on Zoom for a few more seconds. If you're still there and want to ask some questions, you're most welcome to do that.
Okay. Thank you.
Thank you so much, sir. Thank you.
Bye-bye.
Bye-bye. We'll meet again soon. Thank you.